Okay, good morning students again. Today let us discuss about the classical genetics. If you have any doubt means after the class you can interact with me. Are you hear me first of all? Are you able to see this one in the picture? Students, are you able to see this picture in the monitor?
Yes, ma'am.
Yes, ma'am.
Okay, let us
Yes,
continue.
ma'am.
Okay, ma, let us continue now. Okay. Let us start with the topic genetics. So the genetics is a branch of biology that deals with the mechanism of transmission of characters from parents to offsprings. This is one definition, and another definition is that genetics is the science of heredity and variation. Here you can see the picture. So this is actually the parents. The characters are transferred from the parents to the offsprings. So this is about the definition. Then next, this is actually the scientist William Batsman. So he is a British biologist and then Mendelian scientist also. He coined the term genetics in 1906. And the next is the, next let us see the types of the genetics. 4 types of genetics are there. Classical genetics, molecular genetics, population genetics, and quantitative genetics. First category is the classical genetics. It deals with the transmission of genes from parents to offsprings. Here you can see the picture. So this side one is the pistil. It is actually the female parent, and here it is a pollen. This is the male parent. So the offsprings automatically they can get the color of the parents. So the characters are transmitted from the parents to the offsprings. So that is the first category. And then second is the molecular genetics. So the molecular genetics deals with the molecular structure of a gene and its function. So last year you studied about the structure of a DNA. So the same way here you can see the chromosome structure and then structure of a gene and then the presence of the position. So like that you can study it in the molecular genetics. So the next category is the population genetics. So the population genetics actually deals with the hereditary of a traits in a groups of individuals where the traits are determined by few genes. Here you can see a group of insects. All the insects are genetically, they are same, but the external colors are different here. So this is only due to the presence of, it is determined by few genes. So this we can call it as a population genetics. So the next category is the quantitative genetics. The name itself you can know. So the quantitative means the number. So that it deals with the traits of groups of individuals where the traits are determined by many genes. Here in this picture you can see, so if the people gets a 6 genes, that is the number is high means that person will become a black color. So if they are having a 5 genes means somewhat brown color in nature. And then if the 4 genes are present means it is a light brown. And the next 3 means more than light and 2 very light and 1 is white. So this is due to the presence of a quantitative. The number of gene varies means it determine the number also will be determined. And then next, and one more thing you have to see, there is the same, the population genetics and then quantitative genetics are somewhat same, but here the population genetics actually the traits are determined only by few genes, but the quantitative genetics actually it is determined by many genes. Okay. Next, let us see about the variation. So the definition for variation is that variation refers to the differences in the characteristics of organism belonging to the same species or among the different species. Here you can see a group of girls, but all these faces are different because they are showing the variation in their faces. So the variation refers to the differences in the characteristics of organism. So these all are female, but their faces are different. Same way, belonging to the, it may be belonging to the same species or among the different species. Okay. Next, let us see about the differences between a discontinuous variation and then continuous variation. This is an important 5-mark question also. So before seeing the points, let us see this picture first of all. So here you can see the earlobes. So they here the earlobes is a attached earlobe and here it is an detached earlobe. The earlobe is attached with that skin. Okay, but here it is not attached. Here it is an brother and sisters. So all are different. But here this side you see this picture. So different colored persons are present here. Here this side dark colored person and then somewhat brown and a medium color and a light slightly white and then somewhat okay. This is a pure white. Now let us see the differences in these points. First, the discontinuous, the name itself, it is not a continuous one, and then continuous variation is a continuous. So it is happening a continuous one. The first point is discontinuous variation controlled purely by one or two genes. But here it is controlled by many genes. And then second one, no influence of external factors. Suppose this attached or detached earlobes, it is not determined by the external character, only the gene present within the person itself, they are having the character. So they are having this earlobe differences and also the faces are the brothers and sisters also. But here you can see the environment directly influence the phenotypes. Here even the white color people also, white color skin people also, if they exposed in sunlight means automatically their external skin color will be changed. So this environment can be influenced or the environment can be change the phenotype. So this is the meaning of that point. And then third is the individuals show limited variations without any intermediate form. You can see one, it is an attached or it is detached. In between there is no differences. But here you can see pure white color, pure dark color, but these all are in between the intermediate form. But here we are having intermediate colors and intermediate form, but here there is no intermediate form. This is the meaning of this one. So the individuals show a high degree of variation with many intermediate forms. And then no overlapping of phenotypes, only the extreme phenotypes are noticed. And these 2 points are same. If there is no intermediate means definitely there is no overlapping of phenotypes, only the extreme phenotypes are noticed. But here the phenotypes overlap and there is a complete gradation of phenotypes from one extreme to another without any interruption. Here this is also the example. The phenotype one, it may be extreme dark or extreme white in color without any intermediate. Here many intermediate forms are here. And it is also called as a qualitative inheritance. Qualitative, you know the meaning. So it is also called as a quantitative inheritance. So these are the main differences between the discontinuous variation and continuous variation. This is an important 5-mark question and repeatedly asking question also. And next, let us see about the importance of variation. So why the variation must be there? Because it has to prepare the individuals better fitted in the struggle for existence. Because when you take the plants means, so we consider plants, so the individuals has to be a struggle for existence, and it helps the individuals to adapt themselves to the changing environment. If the variation is there means then only that plant can be changed along with the environment. And next, it provides the genetic material for natural selection. For natural selection itself, they can provide variation is there means they can only produce the genetical materials. and then raw materials for evolution is available. And then next, it helps the plant breeders to enhance the yield, growth, and resistance in plants. So these are the importance of variation. This is also important question. And then next, let us see about our great scientist Grigor Mendel. So you all know he is the father of genetics. So he born on 22nd July 1822 in Austria. He is a mathematician and philosophist, and he worked as a priest as an Augustine St. Thomas Monastery, and he worked as a secondary school teacher also. From 1856 to 1863, he did his historical experiments by hybridization experiment on pea plant, that is the Pisum sativum plant, Botany Tavaram, and then he presented his paper entitled as an experiments of plant hybridization in the meeting of Natural Science of Society in Rome in 1866. He died in the year of 6th January 1844. And we all are celebrating him as a father of genetics. And this is his garden. He is doing his experiment. Why he is choosing a Pisum sativum plant for his research? Because this is an important 2-mark question. Because this Pisum sativum plants is an annual plant. You already know what is annual. The plant will grow and die and giving the yield, and they will die within 1 year. So that type of plant is called an annual plant. And then distinct contrasting characters. This plant is having many contrasting characters. And fourth is the self-fertilizing plant. And next, the flowers are large sized. So this is easy for him to do his research work. Okay. And then next, and then this tabulation, it's already given in your book. So why again, we don't want to waste our time. You can refer your book. Everything is clear in this picture. So round yellow means that color and part shape, that is structure you can see. Okay. And the next, we can move to the next one. The reasons for Mendel's success, why he is success in his work means, so he is the first person doing the research work in the genetics. So he is being a mathematician. He is applying his mathematical and statistical data, and then he can get a accurate results from that experiment, and he can use the large samples also. And the crosses, he can made many crosses, and then the selection may be a pure breeding plant he is using for his experiments, so that he can get a good result. And then selection of the characters are highly contrast, and studied the inheritance of only one character at a time, so there is no overlapping at all. And then fortunately, the genes for the selected traits are located on separately, that we have to see the next one. This is one. So why his work is a success means the location of the genes. Last year we studied the structure of the chromosome, and I already told one name, that is the locus. The locus means the place of a gene which located in the chromosome is called the locus. So he is choosing a 7 pair of contrasting chromosome for this experiment. So let us see the location of the genes of the 7 characters. So if you are taking the first chromosome means, you can see that tip, that is the place of the locus. If it is a tip means it is a terminal, that and all we already studied. So that flower color, and then here the seed color in the first chromosome, and the second there is nothing. Third also there is nothing. And then fourth, he is having a selected part shape is a locus is present, and then plant height, and then flower position, and then here you can see the pod color, and the sixth one nothing, and then seventh is the sheet shape. So this is actually he is choosing these 7 pairs of contrasting characters. All these characters are having a different locus because it is not overlapping with each other. This is the main reason for his success. Each are present in different places, so he can easily choose the locus for his experiment. So this is a structure for that one. Next one, these 3 scientists are very important scientists. these three persons are rediscover the Mendel's publications. The first one is the Hugo Duveris, Dutch botanist. He belongs to Holland. In our book, it is given as a Holland. Okay. So, and then next here it is an Carl French Horens, German botanist, and then Erich von Tesmark. He is an Austrian agronomist. So these three persons only, after Mendel died, they took the experiments and they did so many researches. So these three persons are very important, and the name of these three persons and their country is often repeatedly asking 2-mark question. Very important 2-mark question. Okay, next let us see the terms related to the plant hybridization and genetics. First, let us see the self-fertilization. So you already know what is self-fertilization. The fusion of pollen and egg of the same plant is called the self-fertilization. In this picture, you can see this is actually the Pisum sativum plant. That is the a pea plant. So this anther, this yellow color anther actually fell into the stigma of the same plant. So the fusion of the anther to the stigma of the same plant is called the self-fertilization. And the next is a pure line breed or the true breeding line. So what is pure line breeding means? He is choosing one plant means he is selecting a plant as a parent means that parent must be a pure breeding plant. So how can we determine that plant is pure breeding plant means? So repeatedly he is taking that parent plant and doing a self-fertilization. So after the 8th or 9th generation, we are getting the same trait means that plant is considered to be a pure breeding because there is no mixture. If there is any mixture of genes in the parent plant means automatically in the second or third generation, automatically that mixture character will be established. But here, after so many breedings of self-fertilization, here we are going to get a pure plant only. So this we can call it as a pure line breeding. Okay, next let us see about how he is doing that breeding. Breeding means that is the cross fertilization, and also how he is doing the self fertilization. Even in this diagram is given in your book also. First you have to know the important term emasculation. So what is emasculation means? The process of removing anthers from the plant is called the emasculation. So this process, here you see the pictures. Within the picture, one scissor is there. With the help of the scissor, we are just remove the anthers alone from that one. So this process is called the emasculation. So after removing the anthers from the plant, next what we have to do means this is called the bagging. So this process of covering the emasculated means already removed anthers from the plant. So the process of covering the emasculated flower with a butter paper or polythene bags to prevent the cross-pollination with the undesired pollen. So why here we are bagging the plant means once we remove the anthers means next we have to insert the desired character of anthers into the stigma. Then only we can get the result what we are going to expect. So to prevent that, sometimes the cross-pollination, the unwanted pollen may be shed on the stigma. So to avoid that only, after emasculation, immediately we have to bagging the plant. Okay. Okay, next we have to see the terms related to the genetics. First, you already know what is genes. So the genes are the portions of DNA which acts as the physical and functional unit of heredity. This is a definition, important definition also. So this is a portion of DNA. So last year you studied the structure of DNA. So in that structure of DNA, the genes are situated which acts as a physical and functional unit of heredity. Here you can see that capital G, capital R and small s, small t, and these all are, we can call it as a genes. Next, locus. I already mentioned here also the place of location of genes on a chromosome is called the locus. So here this is the locus of the gene. So the gene G is present in this locus. So this green place is called one locus, and then here this R is the another locus that gene is present R. So this is the same way. And next you have to know the term that is the alleles. The alleles, the various forms of a gene are called the alleles. The various form, not same the form. Here you can see the small t, this is one gene, and here it is a capital T. So together we can call a different we can call it as alleles. Okay, I think you can able to follow. And the next, let us see about the terms related to the plant hybridization and genetics. First, you have to know about the homozygous condition and heterozygous condition. Homozygous refers to the condition of chromosomes having a similar alleles of a gene. So here, actually, here in these two chromosomes, here you can see the capital A and capital A. So both genes are same. So both alleles are same. So this condition we can call it as a homozygous. And heterozygous means heterozygous refers to the condition of chromosomes having a dissimilar alleles of a gene. So here capital A allele and small a allele. So if we are having a dissimilar character means we can call it as a heterozygous condition. Okay. And then next, the terms related to the genetics. First you have to know the dominant allele and recessive allele. So what is dominant character means? So the character which is expressed in the F1 generation is called the dominant allele. The character which is not expressed in the F1 generation is called the recessive allele or recessive character. Okay, so here you have to mention the dominant character as an capital, that is a capital letter, that is the uppercase alphabets, and then recessive allele is denoted as an lowercase alphabets in this picture. And dominant allele is the allele that most or overrides the effect of another allele of the same gene. Hence, the expresses its phenotypes in both homozygous and heterozygous condition. And one more character that a dominant gene is having, the dominancy can be expressed if it is an homozygous condition or it is a heterozygous condition. For example, you can see here it is a dominant allele A and here it is a recessive allele A. But here we are having the different alleles. This is the homozygous dominant capital A. Here the same dominant character expressed. Here the heterozygous condition, that one allele is capital A and another allele is small a. So this is a heterozygous condition. And here also one is small a and another is a capital A. This is heterozygous condition. But whatever it may be, if it is a capital allele means capital allele A is present means automatically these three are expressed the character of the dominant. But the recessive can be expressed only in the homozygous condition, not in the heterozygous condition. Here also small a present, here also small a present, but their character is not able to express when the capital A is along with them. Okay, I think you can be able to understand. Shall we move to the next one? And here the terms related to genetics. We have to note that genotype and then phenotype. This is two important words. The genetic constitution of an organism. The genetic, here you can see, if you are saying, by seeing the picture itself, you can see this is a female, and seeing the picture itself, you can see this is a male. But this is actually called the phenotype. The phenotype is the observable character of an organism. So by observing itself, you can see this is a female, and observing itself, you can see this is a male. But when you see the genetic constitution, If it is an X means automatically it is a female. If it is an Y means it is a male. So the genetic constitution of an organism is called an XX, XY is called the genotype. So you have to know the genotype and phenotype. So externally you can see means keep it in your mind. These are the important terms. You have to always keep it in your mind. So external observable characters, you can call it as a phenotype, and then genetical characters are called the genotype. Okay. Next, let us see the monohybrid cross. So what is monohybrid cross means? Mono means you already know a single one. Okay. So the monohybrid is a cross between the two genetically different plant of a species where the inheritance of only one trait is considered. So if we want to easily remember means monohybrid means by Mendel choosing only one contrasting character for his experiment is called a monohybrid cross. So here he is choosing only one character, that is a height. So he is choosing two different parents. So first parent is having a tall plant. You can see this Pisum sativum plant is a tall plant. This is a capital T, capital T. And then here this is a Pisum sativum small plant, that's a dwarf. We should not call it as a small. So here we have to mention a dwarf plant. This is a small Pisum sativum plant. So here the alleles capital T, you can see small t alleles, but it will be from the alleles actually present within one gamete. At the time of gametic formation, they segregate into a single gamete, that only it will be reunite again. Okay. So this is actually there's a capital T from this gamete and small t from this one combined together, and we can get it as a capital T and small t. So in the F1 generation, what is F1 generation means? First filial generation. So in the first filial generation, all the genotypes we can get there. What we can get? Tall plant. So the tall plant, capital T, small t also, in heterozygous condition, we can get this tall plant. So F1. So again, what we have to do means for F2, this F1 plant has to be made. This is called a self-fertilization. So self-crossing we can call it. So self-crossing we have to do in the F1. Then here we can get the F2. So in the F2, here we have to put the gametes like this, capital T, small t, capital T, small t. Then we can get the traits like this. You see here. Okay, here you can see the tall plant. This is also tall plant, and this is also tall plant, and this is a dwarf plant. I already told the observable characters. If you are observing these three tall and one dwarf, so this is actually, you can call it as a phenotype. So how many tall plants? 3 tall plants is to 1. So the phenotypic ratio is the 3:1. Okay. The same way, if you are seeing the genetic constitution means genotype is a capital T, capital T, capital T, small t, small t, small t. So the capital T is a homozygous tall is 1, and then heterozygous tall is 2, capital T, small t, and then capital T, small t, 2, and then small t is actually a heterozygous condition. Okay. So this is actually, this person is Punnett. You already know the Punnett square. So the square itself named after his name. So the Punnett square, or we can call it as a checkerboard. So this checkerboard, why we are using this Punnett square means, Punnett square is a square diagram, helps to calculate the probability of possible genotypes of offsprings in a genetic cross. So how many possibilities, that probability easily we can calculate within this Punnett square. Okay. So one side we have to consider as a female, and another side we have to consider as a male. So we have to take a gamete, capital T, small t, capital T, small t. So together here we can get capital T, homozygous tall, and then capital T, small t, heterozygous tall, and here capital T, small t, heterozygous tall, small t, this is homozygous dwarf. Okay. So this is about the Punnett square. And then next, let us see about the reciprocal cross. So what is reciprocal cross means if a cross is performed by a reversing the sex of a individual, such a crosses are called a reciprocal cross. So here what we are going to do means that sexes are going to be a reversed process. You just before seeing the description, you see the diagram here. So this is a cross one and this is a cross two, reciprocal cross. In this cross one, first one, we have to consider the tall plant as a male and then dwarf plant as a female. Okay, yeah. So the sexes are different, but we are considering the male plant as a tall and then female plant as a dwarf. So here we are doing the cross. So in the F1 progeny, we are getting all tall plants. Okay, and then you see here in the class 2, that tall plant, here we are considering female as a tall plant and a male as a dwarf plant. Okay, and F1 here also we are getting F1 progenies are all tall plant. So here what is reciprocal cross means you may consider as a male or female as a tall or dwarf. Okay, yeah. The dominant or recessive characters are not determined by the sexes. So this is an individual character of an organism. The individual character of an organism does not determine by the sex. So this you can easily understand by the cross. So that is only given here. The example class 1 male as a you already know. Okay, so we don't want to waste the time. So why we have to do the reciprocal crosses? The objective of performing the reciprocal crosses is to identify whether the sex of the parents has any influence of the inheritance of the traits. Why we are doing this male and female, why we have to change means, suppose if we change a male or female means whether the character is going to change or not. So for that confirmation only we are doing this reciprocal crosses. Okay, I think are you able to understand the reciprocal crosses? Okay, shall we move to the next one? Next is the back cross. So what is back cross means? The cross between the F1 hybrid with any one of its parental genotype is called the back cross. Okay. So first here also we are getting that parental genome capital T and small t. In the F1 we are getting the capital T and small t. Okay. Yeah. So the capital T small t is again back cross with any one of the parent. The parent may be a dominant if it is crossed with, I will explain here. You see here in the F1, so here this is one parent, a tall parent, this is a dwarf parent. In the F1 we are getting the capital T small t. So this capital T small t has to be again crossed with the dominant parent, or again it has to be crossed with the recessive parent. So this is called actually the back cross. Okay, so this is a, if we crossed with a capital T means we can call it as a dominant back cross, and if we crossed with a recessive means we can call it as a recessive back cross, or other ways we can call it as a test cross. Here you can see if it is a dominant back cross means the F1 hybrid capital T small t is crossed with the dominant one capital T. So here again the gametes are segregated like a capital T small t and a capital T small t. So we have to keep it in the Punnett square. So one side considered as a female and another is considered as a male. So capital T small t, here capital T and capital T. So this is a tall plant and here tall. So all the traits are tall plants. So this is a 100% tall plant. Okay. And then here let us see the recessive back cross. This F1, the recessive back cross, the F1 hybrid capital T small t is crossed with the recessive parent, that is the small t. So the gametes capital T small t, here the small t. If we are keep it in the Punnett square means here it is considered as a female and this is considered as a male. So the capital T small t and then small t small t. So here we are getting these two tall and here these two tall and this is heterozygous draw and this is heterozygous draw. Here we are getting equally, that is a 50 percentage tall and then 50 percentage of them is draw. So equally we are getting in the recessive back cross, but here in dominant back cross we are getting all 100 percentage of a tall plant. And then next one, okay, next one. So why we have to you can see here all the progenies of a dominant back cross will be dominant only, no recessive trait will be occur. Okay, and then here the progenies of the recessive and then it will be cross will be so equally, the terms are equally 50 and then 50. Next, the what is the objectives of the test cross means the test cross is mostly done to identify whether the genotype of an individual is homozygous or heterozygous. Okay, the cross why we are doing means we have to understand whether it is a homozygous condition or heterozygous, because in the F2 only we can identify that a trait is a homozygous or heterozygous. So if the result of a test cross in an equal proportion, both dominant and recessive, then certainly the F1 individual is an heterozygous. Here it is a heterozygous, so we can get a equal proportion. So if the result of a test cross shows only the dominant type, then all the F1 individuals are homozygous dominant. Maybe it may be a homozygous dominant or it may be a heterozygous dominant. So we are getting a all tall plants. Okay, so these are the objectives of the test cross. And next, let us see the dihybrid cross. So the as I already mentioned, the monohybrid means Mendel chooses only one contrasting character means this is a monohybrid cross. But if he is choosing a two contrasting character means this is called the dihybrid cross. So the dihybrid cross is a cross between two genetically different plant of a species where the inheritance of two traits is considered. It involves the inheritance of two genes of each having the two alleles. Okay, so you have to see here. Here you can see this is actually the parent plant. But before seeing this, why this round yellow in this picture you can see he is choosing the Pisum sativum seed. I think you already have it in your mind. So the Pisum sativum seed, actually this is a round shape and here it is a wrinkled shape and this is yellow in color and this is a green in color. So all Pisum sativum plants maximum they are round in color only. But why we are getting the wrinkled shape means I have to explain. So you just listen. So here you can see the traits. This is a one character. This is the round yellow and the next is the round green and the next is the wrinkled yellow and then wrinkled green. So why that round yellow and then round that color, why that shape is different, I will explain. So in that actually the Pisum sativum plant is having a dominant, if it is having a dominant allele, capital R, R means actually it is a round shape. So the if it is having a homozygous capital R means it consists of a active enzymes that is called a SBE. Okay, yeah. So that active enzyme that it is consist of a linear unbranched amylase. Actually, it is not in that PPT. You just listen and then we can follow. So the linear unbranched amylase is converted into a branched. The The linear branched amylase is converted into a branched starch amylopectin. that so we can get the round piece. So if it is having a recessive allele, small r means inactive enzyme, that linear unbranched starch amylase, there is no conversion. So the linear unbranched amylase will give the wrinkled color, wrinkled shape. Okay, now let us see here. It is a, so here it is a round yellow, here it is and round yellow and then wrinkled green. So that genome we can see capital R round, capital Y yellow color, small r wrinkled, and small y green color. So we can get a gamete capital R capital Y, small y. So in the F1, all the F1 progenies we are getting what it is, a round yellow we are getting. So again we have to self cross the capital R, capital Y, small y. So the all progenies, this is actually the dihybrid cross. Okay, self cross F2. Let us see the next, that Punnett square. So this we have to keep it in the Punnett square. So we have to separate the gametes from here. So the capital R, capital Y, capital R, small y, and then small r, capital Y, and small r, capital Y. Here how the gametes are separating means individually you have to split into a capital R, small r, and then you have to cover this capital Y, small y, and first the R joined with this R and this one here, and this R and this small y here, and second you have to small r and a capital Y here, and then small r, small y here. The same gametes you have to put this side also. So again, you have to combine these two. So capital R, capital Y, and these genomes easily together you can write. So here you can get the 9 round, the phenotypes, the phenotypic ratio. Here, how many round yellow colors are there? You can see the 9 round yellow, and then 3 round green, 1, 2, and 3, and then here wrinkled yellow, 1, 2, and 3, and then wrinkled green, here it is 1. Okay, so together you are getting the phenotypic ratio 9 is to 3 is to 3 is to 1. So if it is a genetic constitution, well, actually the genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So how can you easily remember this means, so actually this is a 9-digit number. So in the middle, first you have to put 4, and then next to 4, 2 2s are 4. So here you have to put 1 2, and then next to 2. So middle 4 and 2 2. Next you have to put 1 1, and the next 2 2, and again 1 and 1. So like this you can keep. Okay, yeah. In this way easily you can remember. So that genotypic ratio. So another one. So easily after putting the cross, whether is it correct or not, easily you can draw the structure like this. You have to draw the structure. So you see, you have to put a triangle like this. Okay, yeah? So you have to put a triangle like this. So you see here, I will draw the structure now. Wait here. See here, you can see, we draw a structure like this. If you draw a triangle like this, so exactly it is coming, this is a big triangle. So the big triangle actually is a round yellow, and then second triangle from here, and then third triangle. Okay, here the last one. So if you are noticing means the large big triangle, all the triangles actually it is going through the round yellow, and this second triangle only the tip of the second triangle is the round green, and then third the small triangle tip also again this is called the wrinkled yellow. Last the single one it is actually a wrinkled green. This whether you can check whether your gametes are correct or not. Okay, so if it is an so if it is a correct means you can draw the external structure. Okay, yeah. Okay, let us see the next one. So this is actually dihybrid cross, and then this is an important 5-mark question also. So here this is a dihybrid test cross. So the dihybrid test cross, the phenotypes and the genotypes. The round yellow, the F1 is a capital R small r and then capital Y small y. That wrinkled green is an small r and small y small y. So it is crossed with the recessive parent. So again we are getting a genotype here, that is a round yellow, round green, wrinkled yellow and wrinkled green. So this is 1 is to 1 is to 1 is to 1. So if the test cross is coming like this 1 is to 1 is to 1 is to 1 instead of 9 is to 3 is to 3 is to 1. So this progeny, this is a pure breeding plant. So to check the pure breeding nature of the character, here we are doing the test cross. So this is a again dihybrid cross. The sheet shape with the two form round and then wrinkled. Here it is in yellow and then green. So this is the genotypic ratio and this is the phenotypic ratio. Okay, and then next Mendelian law of inheritance. So that is the law students, please don't draw now. You listen the class. Afterwards, you can draw. So the next is the Mendelian law of inheritance. This is law of dominance, law of segregation or law of purity of gametes, and the law of independent assortment. Okay. So what is law of dominance? I already told. The character which is expressed in the F1 generation is called a dominant character. So here it is a capital R, here it is small r, pure breeding parents only. But in the F1 we are getting this capital R color. So this color is considered to be a dominant. So for the dominant we are giving a capital R, and then here we are not giving the color of the white. So this is actually the a small alphabets of a small one. Okay, yeah? And this is a pure breeding variety and the law of segregations. So within a allele, within a gamete, two alleles are there. This is a capital T, and then capital T, small t. So capital T, and then capital T, small t, and then next one, there's a gametes are again, they are separated into two. That is the capital T and the capital T. Here the capital T, small t. I already told before the gametic formation, the alleles present in the gametes, they segregate here. So that is called the law of segregation in an organism at the time of gamete formation. The allelic pair of a gene segregate and as a result, each gamete will have one allele of a gene. Okay. And the next is principles of segregation. So the principles. Okay, ma. The last one, the principles of segregation. Last year we saw the same, that is mitosis and meiosis. The same way here, the law of independent assortment. So within a gamete, so many alleles are there. So at the time of gametic formation, they are segregated, they are assorted. The characters are not mixing or blending with each other. Okay, yeah. So this is law of independent assortment. Last year we draw the structure of the meiosis. Okay, so from this we can conclude this session. We can continue in the next class. So if you have any doubt means afterwards you can contact me. Thank you, students. Okay, so may I know your feedback, please? Hello.